Hi, good afternoon to everybody. Many thanks for joining this webinar. Guy Hendrik speaking from Avia-GIS in Zuursel, Belgium. This is the fifth webinar in our series, the second of this year. Before we start, just the classic practical information is that we will record this webinar and make it public on YouTube afterwards, so that everybody can follow even afterwards. This is specifically the case now because there still are some people on Easter holidays. During the webinar, participants are muted, but can ask questions anytime using Q&A button at the top left-hand side of your screen. And at the end of the talk, I will moderate the questions. Well, our speaker today, you all know, I think. It is Isra de Blauwe from the Tropical Institute in Antwerp. She holds a PhD in biology. She was working on chimps and just explained us it was a lot of scatological work she was involved with, but interesting. She was focusing on, you know, what kind of insects these apes are eating. But then she turned to the real stuff, working with mosquitoes, something we all prefer to do. Anyway, today she will present part of her work reflecting on how to best conduct national surveys for monitoring invasive species of mosquitoes. We've been working together on that for quite a while, and she definitely has a lot of experience in this and will use Belgium as a case study. We are in Belgium. Avia-GIS is also involved in this kind of work, but mainly from the control side. Actually, that's one of the rare control activities we do. We are exploring together with the Walloon government how this issue of invasive mosquitoes can be tackled in a practical way, which is not easy. But for now, Isra is going to talk us on how VECMAP can be used for surveillance of invasive Aedes mosquitoes in Belgium. So the floor is yours, Isra. Enjoy your talk.
Thank you. So yeah, welcome everybody. I will present to you the use of VECMAP software in our invasive Aedes surveillance in Belgium. Well, I think you already all know what are invasive mosquitoes, but I didn't know who was the public, so I'm just gonna shortly introduce them to you. So there's 4 invasive Aedes mosquitoes that are present in Europe today, of which 2 are important vectors of several pathogens. You have the Aedes aegypti, or the yellow fever mosquito, which is a well-known vector of yellow fever, dengue, Zika, and chikungunya. And you also have the Aedes albopictus, so Aedes Asian tiger mosquito, which transmits dengue, Zika, chikungunya virus, and also the heartworm Diarrhoefilaria in the field, and many other viruses, and is also a competent vector in the lab of yellow fever and West Nile virus. Then other invasive mosquitoes are Aedes japonicus, the Asian bush mosquito, and the 4th one is Aedes coreicus. And they are less involved in the pathogen transmission in the field, but they're competent to transmit some pathogens in the lab too. Now, the distribution of these mosquitoes in Europe, you see here the ECDC distribution maps, the recent ones of 2022. They're all established in Europe. You see the red areas. The yellow areas indicate introduced specimens. 3 of the specimens have been found in Belgium. 3 of the species have been found in Belgium already. Aedes albopictus, Aedes japonicus, and Aedes coreicus. Aedes albopictus is approaching the Belgian border. You see they're getting very close to the Belgian border. Also, Aedes japonicus is very close. Yeah, and Aedes coreicus is already present also. So, but Aedes albopictus is not yet established in Belgium that we know. However, every year we record them as introduced specimens. Aedes japonicus and Aedes japonicus is well established in our in Belgium, in Namur. We have been trying to control it, and it still stays a small population, but it's not gone yet, and probably will not be gone anymore. And it is also introduced along the border with Germany. We found them in Eupen and also a bit higher in the Limburg. Just introductions. But for Aedes coreicus, the species is well established also in Flanders and Limburg. But there the population is also still present and now is also being controlled. However, probably will not be able to eliminate it anymore. So these species were all introduced through the transportation of goods, mainly used tires and lucky bamboo for Albopictus, but recently also with ground traffic from neighboring countries. Now, the aim of the surveillance project in Belgium is to detect as soon as possible these invasive Aedes species to be able to delay their establishment or the spread. Therefore, several import pathways were monitored longitudinally, the so-called point of entries. And here you see the map of Belgium with the point of entries that are monitored during the MIMO project, that was between 2017 and 19, and during the MIMO Plus project in 2020. They include ports, airports, flower auctions, wholesale markets, used tire and lucky bamboo import companies, also parking lots along highways and industrial areas. We also included, to detect the spread of Aedes japonicus from Germany, we also included cemeteries and an allotment garden along the border. Now, how did we monitor these invasive Aedes species? We used several traps that attract the host-seeking females using the CO2 and lure, such as the BG Sentinel trap and the mosquito magnet trap. We also used traps attracting gravid females using water or an infusion, such as the Frommer updraft gravid trap and the BG cat trap. The same principle was used to collect eggs using oviposition traps with water or an infusion and the floating substrate. And finally, we also actively collected larvae in different potential larval breeding sites, so using aquarium nets and sometimes a dipper. And for all the data we collected and for the data management, we used the VECMAP software. Now, in this presentation, we will focus on the data management of the Aedes monitoring. So we collected field sampling data, also identification data, both morphological and molecular data. And of these specimens, we sometimes selected samples to be added to the molecular and the morphological reference collections. We made use of the VECMAP software for the real-time project monitoring, specifically to collect our field data and the morphological identification data. Before the start of a project, the software can also be used to set up the project design. But we have created our own sampling strategy, which was based on points of entry that were selected, and also the risk scenarios, if it's introduced or an established invasive Aedes species. This determined the activities and the frequencies that we needed to do. So it was mainly longitudinal study. Also based on the available staff, we based the project design. So this monitoring scheme that we made, we planned it in a Google Calendar schedule, and this info was already added to the VECMAP system in advance by uploading the CSV file with pre-filled data already. So how does it practically work? We have, VECMAP has 3 components, and we use 2 of them. First, we use the web component to manage the project by setting up a sampling scheme and produce the forms that need to be filled in by technicians to collect the data in the field and also for the lab. We also use the web component to enter our morphological identification results. And as the data is a longitudinal collection, it's longitudinally collected, we fixed with fixed locations, mainly points of entry, and a fixed sampling strategy based on scenarios, risk scenarios. We had a specific data structure that was created in VECMAP with a traceable and unique labeling system. Second, for collecting the field data, we used the application on the smartphone. So technicians and also some local partners with who we work in the field filled in the forms directly in the field. The third component, the desktop component, which incorporates GIS and modeling tools, but also can be used to track status of the project. We did not use it a lot yet because the fieldwork is daily coordinated by one team, and we do not do any modeling yet with the desktop function. So to set up the database for the field collection, we created 3 levels in the web component. The first level are the locations, so the points of entry to be surveyed. The second level that was created is for the traps that are placed at the point of entry, or also for the potential breeding sites that are checked or investigated at each point of entry. Then each point of entry has, of course, several traps and PBS. And all trap, all the trap and PBS info is stored at the second level. For the final and third level, we created this for the sampling events. So at each point of entry, several sampling events took place with the different traps or from different PBS, potential breeding sites. And all information of the sampling event is recorded at this level. So on the smartphone, you can also see the 3 levels you see here. You can choose the levels. Now, in the first level, the general information on the point of entries where we are monitored is recorded. So each point of entry gets a code. We give them a code. It's a 2-digit code linked to the name. So here you see, for example, VP, which is a used tire import company. And we also have E6, which is a parking lot. And at this level, all information about the point of entry is stored. So the information on the type of the company, which is often related to the import pathway of the invasive Aedes, also the address information, general coordinates, contact details, and specific remarks for the field team. For example, is registration necessary at the company or not? And in this way, if a technician needs to go to a location that he didn't go before, most of the important information is available in the smartphone. Here you see how it looks like in the VECMAP web component. So this is the metadata, the standard metadata you can choose in the beginning when you set up the project. We choose for the name, so you have the code, and then you can put the coordinates there and you see it on the map. But if you want more information, like we want, address and stuff, then you need to make your own layout, which we did in this way. We added a field for each of the specific data we wanted. So the type, the street, postcode, contact number 1, contact 2, etc. You also have a preview of how it looks like on the smartphone. And as in the surveillance projects, the locations are fixed, point of entries are fixed. We added this info in advance by uploading a text or a CSV file. But it's also possible to create a new point of entry in the field if you want to. Then at each point of entry, we have multiple traps or multiple potential breeding sites that are investigated. So in the second level, the trap and the PBS info is recorded. Each trap or a potential breeding site has its own code. The trap code starts, for example, with the letter of the trap. BG Sentinel, for example, B002 is BG Sentinel number 2. And each potential breeding site also gets a code, for example, with the point of entry code first, VP, and then you have an L referring to larvae and a 3-digit number. Also the type of trap, the BG Sentinel mosquito magnet, or the type of PBS, for example, if it's a plastic sheet or tire is recorded. And then also coordinates of the trap or of the breeding site can be recorded in the field with a smartphone. But sometimes with precision depends a bit on the phone type. So if you want precise mapping, very detailed mapping, you need sometimes to adjust it afterwards. With a smartphone, we also take pictures where the trap is located, or pictures from the breeding site, which is checked. So here you see again the web component, how it looks like. You have the trap code. You have also the coordinates of the trap and on the map. And then we also made a layout for this with the fields that we want. Here we have the type of trap, remarks that you can add, and we added 3 photos. And as a sampling strategy is fixed and we have already an idea of which number of traps is known per point of entry, we already also upload a text or CSV file with these codes and types. The photos, of course, are taken in the field and the coordinates, but you can upload already some information in advance that you don't lose time in the field. So here you can see how it looks like in the web component. For example, we have here an oviposition trap. We have a picture we have 3 different views of the ovi trap. So we choose 3 pictures, one of close by with the code on the trap, how it's positioned, one a bit further away, and then one far away, sometimes with someone pointing where the trap is located. So it makes it easier to find back the trap in the field, especially when someone new has to go in the field. And also sometimes different containers belong to the same potential breeding site code. For example, you have a pile of tires in the one corner, in the other corner. It's one code, but you take sometimes pictures from the different piles or from different plastic containers. You can add 3 pictures. So if the trap location or the potential breeding site is the same the next year, you can also upload already existing pictures through the web component. So not in the field, but already in the web app. But it's easier also when you duplicate the project with all the information, including the pictures of, for example, the first level of point of entry and the second level. And this is a recent new feature in the web component. Yeah. So in the third level then, we have the results of the sampling event that are entered in the database. So each sampling event also gets a unique code. We use a code starting with the project letter, the project code. So M is from memo, 19 is from 2019, for example, followed by the point of entry code, the VP, for example, and then the trap code, B022, for example, and then we have also the start date, August 14. Most data is entered with the smartphone in the field, of course. But like the start date and end date of the capture, we plan it already in advance. So that can be entered already also in advance, uploaded. We also entered the collectors who collected the samples, the status of the trap, if it was working or not, falling over, and also the number of potential breeding sites, containers that were sampled. So here also for each of these sampling events, we have the standard metadata. You can choose what you, in the beginning, what you want to see. The map here was not really needed, so next time I will leave it out because coordinates are actually the ones of the potential breeding site in the level 2 or of the trap. So it's not really necessary. However, we have to give it a unique lab identifier, and for which we use the same code as the sampling code. In this way, we can find it back easier in the sampling results afterwards, in the lab results afterwards. You can also make your own layout again, and see how it looks like on the smartphone. And as I say, the sampling strategy is fixed. So, and we know when we go in the field, we upload the text or CSV file with the data in advance. Sometimes also the collectors we know when we already pre-fill it. But the other data, of course, is filled in the field when you go there and collect your samples. At some point of entries, mainly the parking lots, we collaborate with local partners, persons who maintain the roads, for example. And they also they do the trap collection for us during the season. So they send us the substrates of the OV traps by post. And between 2017 and 19, we didn't let them use the mobile application yet, but they recorded data on the paper. Yeah, it's because we also had to get used to the software first ourselves, and then it was easier to give them the paper. And then with the samples, they send us the paper, and we entered the data in VECMAP management system in this memo project. But now in the Memo Plus project in 2020, we wanted to test the use of this assignment function of VECMAP. And therefore, we did a training in VECMAP. We give a training to the local partners at the 2 parking lots. We made a manual, which you see here, a page of it, how they have to fill in the data in the app. We did it in Dutch and French. So, and then they could use it during the season. The advantage is that you can see also on a calendar on the web component if the assignments are completed or not. And if not, we send an email to remind them about the sampling. And the same data was also entered on the paper form. We did it to check if the data on the forms corresponded to the data that was entered in VECMAP. Now, at the end of the season, we evaluated the VECMAP app use by the local partners. As there was a first infrequent use of the app, so they only use it once a month. They needed a lot use of the manual, so they need to look a lot to the manual, and that they lost some time. But I think when they will get used to the app and use it more often, it will go smoother, let's say. And secondly, the data on paper was a bit more accurate than in VECMAP, especially the general info, such as the sampling date, and also the OK status of the trap, although some fields are required to be filled in the app. So we have the required fields. It does not give really a warning when it is not done. So incorporating this warning in VECMAP app would help also local partners to not forget to enter some data in the future. But some of the adaptations have to be done in our creation of the forms. I think this would also solve it. Like now, I use the actual date of the phone to appear, so that would be, it's easier than before when we didn't have this. So it also depends a bit on how you make your own, create the layouts. It's very important to do it in a correct way. Then when you have collected your field data, of course, then you bring samples to the lab, and they will be identified in the lab. And thanks to this unique identifier, which is the same as a sampling code, the results of morphological identification can be entered through the web application. So for each sampling, we have identified mosquito specimens. These are stored by species, sex, life stage separately in tubes. And they receive also a unique ID code. And this is actually the tube code. So the data that is entered is the species, the sex, the life stage, number of specimens, also the executor who identified, and the date of identification. And finally, also the storage box code where it is stored, and info if it was added to the reference collection or molecularly identified. This is also entered in the database of the morphological identification. So we made a specific layout for this. And yeah, this can only be viewed in the web component and not in the app, in the smartphone app. So here you see also how the results of the sampling event in the lab results on the web component look like. It's a list with records, actually the tubes with unique ID code here. Here. And you need to, this overview is also recent. You see also the associated data for each of the codes. It's recent because before you only saw the codes and you had to click on the record to see the data. But now it's really, it's more in Excel file form, which is nicer. You have a nice overview of what you have identified. Only thing is that we still have to fill in each field separately. So maybe in the future, it would be nice to be able to copy the line and then, change only the things you have to change. But this is for the future, I think. And then, so once you have season finished and you want to analyze the data or for reporting, you can download it from the VECMAP web component. You can choose to download the field data or the lab results. Unfortunately, a combination of both is not yet possible, but I hope in the future this will be the case, so that you have the combined information of the sampling and the lab identifications. But when downloading the field data, you can choose the level you also want to download. For example, the level of point of entry, of trap, or of the sampling. You can also download this together. However, when you download this together, the levels are not linked or combined in the Excel, but listed one below the other. So in the end, you have to, depending on how you want your database to look like, you have to do some data management in Excel or R to organize and combine the data. So this is something which still needs some improvement. You can also select the type of the delimiter and the decimal separator. For in Belgium, for example, we use a comma for this decimal separator. Now, next to the morphological identifications, we also have the morphological reference collection and molecular identifications. So some specimens are just stored dry or ethanol in a general collection. Others are selected to be pinned or added to the morphological reference collection, which is hosted at the Royal Belgian Institute of Natural Sciences. And some specimens have missing diagnostic characteristics or exotic, and these are sent immediately for molecular identification or barcoding to BOBCO, which is the barcoding facility for organisms and tissues of policy concern in Belgium. And at the end of each collection year, during MEMO at least, 5% of the specimens were randomly chosen for the molecular identification as a quality check for the morphological identifications. But the molecular database and this morphological reference collection database are not stored in VECMAP. it's not possible like in the lab results that you make 2 levels for this. So it's, yeah, we work with separate Excel databases for this, but it would be nice to be able to do this in the future too. Then you see for the samples that were sent for molecular identification, they're transferred to specific tubes containing a unique QR code, the 2D Smart Scan QR boxes. And the details are entered in a transfer sheet in Excel, you see here, containing the ID code, the sampling code, and morphological identification, and some remarks or instructions. And after identification, molecular identification, Bobco records all results and metadata in their standardized lab book in Excel, ensuring a traceability of each procedure. Now for the morphological reference collection is indicated directly in the lab results part in the VECMAP database. The unique ID code is used as also the unique identifier for the reference collection. And we had also a general collection number for the whole memo project was created. And of some specimens, also high resolution stacking photographs were taken, and this was indicated also in the collection database at Erbins. Now, for all specimens which we generate barcodes, the DNA sequences are submitted to online DNA reference databases in GenBank or BOLT. So here you find a summary of the database structure of the invasive Aedes monitoring in Belgium. It mainly consists of a sampling database with 3 levels and an identification database of 2 levels, which are all linked through a traceable and unique labeling system. And the first 4 levels are stored in VECMAP data management system, while the molecular and the reference collection databases are still separate in Excel. So below you see the different information which is stored at each level, indicating which data was recorded in VECMAP and which data outside VECMAP, mainly in Excel. Now, yeah, to end, I have put here some pros and cons of using VECMAP in field monitoring and laboratory activities. So some working points are that sometimes the app is a bit slow and sometimes has bugs. And the mobile app and the web app could be more user-friendly, which I mean that, like, for example, you need for local part a lot of steps when using this assignments function the way we do. And there's also no warning for these required fields. For the web app, in the lab results, it would be easier to be able to copy, like I said, a record as in Excel. And now you have to fill in all the info separately each time. Then when viewing lab results, it's not possible to see like all the metadata of the sampling event at the same moment. Like you don't know the sampling if it was which trap or which environment. Then when a unique identifier is entered, each sampling event has immediately a lab result. I mean, when you make your sampling event and you copy your code for the lab identifier, it immediately has in the lab results a result already. So there's yes under has a lab result. But this does not mean that it has a record already added. So this is a bit confusing. But these are things that they are working on. So no, also there's no, yeah, like I said, no extra levels possible in the lab results. You have 1 level only. Downloading combined database, so everything together in one database of the total photo database in one, so you have to download each photo separately, is still not possible. And sometimes you have problems with uploading large photo sizes in the web component. It's not possible also, but this is not so important, to open the phone number or email address through the mobile app. You cannot tick on it and go to the app immediately, or the phone or the email account. But in the end, there's a lot of good things too, of course. So entering the data immediately in the mobile app avoids extra work and mistakes. There's the layout forms can be made, it's very flexible. You can really do everything what you want with it. There's compatible with Android and iPhone. There's a fast follow-up in case of error or bug. So if there's something, it's often quickly repaired. There's a possibility to add photos, of course, that's practical in the field. In the map, there's a map in the mobile app with locations where you have to sample. So you can also click on it and then ask the direction to go there. And there's a cloud database, which is everywhere accessible, of course, when you have access to internet. So the advantages outweigh the disadvantages. Okay, I hope everyone understood very well. And if there's any questions, feel free to ask.
Thank you very much, Isra. You can maybe go back to the previous slide, or I mean, for me, it's fine to keep the pros and cons on the screen.
Okay, wait.
Thanks a lot to Isra for this presentation and for her critical analysis or evaluation of VECMAP in this kind of context. Of course, I mean, originally VECMAP was developed for large-scale distribution modeling, working on cross-sectional surveys, which means surveys where you only need to go to one, you need to select a lot of places, and you need to go only once to each place, and afterwards these data are used for spatial modeling. Whereas here, and that was really interesting when starting to work with the Tropical Institute, is that here the focus is more on a longitudinal approach, where we definitely indeed there's still room for significant improvement, and that is what we do with them. And that's why we like to show this as an example where actually users who actually use VECMAP can help us a lot in further improving the software. So at the moment there are no questions. If any of the participants has a specific question regarding what was discussed here, please post it. Otherwise, what I can definitely say is that, as it was said, we are reacting to this kind of questions as fast as we can, and we then actually implement it, and we are looking to ways to do this even faster. So I see that now there is a question in the chat. I can go on to read it. Thanks, Israeli, for demonstrating this longitudinal approach. If your sampling traffic scheme is targeted to invasive Aedes species, what is the rationale for also lab identification of non-target species? That's a specific question to Israel. So what is your rationale for also identifying non-target species in the lab?
Yeah, well, this was only during the MEMO project, actually. So between 2017 and 2020, the MEMO project focused on the exotic mosquitoes, not only Aedes, but also exotic Anopheles at airports and stuff. So we identified everything, and also because it has been a while since there was some notion about the other species in Belgium, the government also wanted us to identify all the other species. So we just identified everything. So I have to say, for the new project now, MEMO Plus, we don't identify the other species anymore. So this is only focusing on Aedes, invasive Aedes. So we only we look at the genera, but we only collect the invasive Aedes and identify them. So it will be a bit less, we will be less using the lab results part, I think, in this new project. So, but still, if I find something special, I will still identify it anyway. So.
Okay, nice. Thanks. Yeah. What impact on the cost do you think? What is the impact on the cost to do the full identification?
Yeah, I think it's still sorting is also a lot of work. So you still have to go through all the mosquitoes anyway. Although, yeah, we also now work mainly with the ovi traps in the last project in the MEMO Plus. So we will not use a lot of adult traps anymore. But I think on the cost, I think it will reduce a little bit the cost of the lab identification, but on the other hand, in these point of entries, most of the time you find the same species. So in the end, you have to count them, of course, and you have to identify them. But if you know it already well, it can go fast too. But it will.
You expect like 20%?
It will cost less, but I don't know if it will make a big difference.
Okay. I mean, do you also?
You have to also not put them in tubes anymore, so that will also lose less time. You just put them in a bunch, and you don't have to do the handling of all the separate species, the separate sexes. So yeah, it could save some time. Yeah.
As you know, ECDC is very much interested into looking at abundance. So there is a question here: do you plan to look at seasonality in density of, for instance, cool excipients in this kind of setting?
For the moment, not. Like I said, we now have a new project which is a lower budget, and we have to also focus on the Aedes, mainly Aedes albopictus. So we will not have the time to really look at all these culex. And also, like I said, we use only oviposition traps at the moment, and larval sampling will be done only when we find something. So there will be much less data than we had in the memo project. Yeah, for now. So maybe in the future this is possible.
Yeah, well, actually, thank you for that answer. This is the kind of issue that with Avia-GIS we're also facing. Is we now are contracted by the southern part of Belgium to do, actually to help them control or plan control about these kind of problems. And then the discussion is very quickly into what is research and what is actual operational, and to reduce the cost, of course, because budgets are cut down everywhere. And that then this kind of added value may be too costly, and indeed that may be the topic of a specific research project. But often in a surveillance program, this may be, well, that the part you need to cut down. So another question is, do you do PCR analysis of individual eggs, or do you work in pools?
We do it individual, but we don't do all the eggs of the whole of the eggs that are on the substrate. We have these polystyrene pieces, and we take from, if we have eggs, we take from each side 5 eggs, and then these eggs are identified separately with PCR. But we don't take all the eggs. So we take sub-sample.
Why did you use for that method?
Because it's costly also to do the barcoding. So we had like a number of that we could do. Yeah. So it is a bit of a wing. And we don't. It's not that we find a lot of eggs of Aedes albopictus, but like for the Japonicus and stuff. But if we have albopictus, it can be that we do some more if we have only this number of eggs on the substrate. But for the Japonicus, we have a lot of eggs, so there we cannot do all the eggs because it costs too much also.
Okay, thank you. Do you want some additional information about this question? Are you happy with the replies? Any other questions? Otherwise, I will definitely would like, of course, to thank Isra for this really nice webinar. Thank you, all participants, for being there again. I've been looking through the list. These are most of you have been following most of the webinars. We're looking forward to the many people who are going to look at the YouTube version or record of this webinar. And we thank, of course, Mathis, who is providing technical backup for making this work. The next webinar will be held on Friday, the 17th of June. We keep them about 8 weeks apart at the usual hour, which is 12 p.m. And it will be about a more technical topic, but very important when talking about spatial modeling. Something, of course, when you take the limited numbers of sampling points like shown today, this is normally standard, not sufficient for spatial modeling, but of course, then we have a temporal analysis options. We're going to talk about pseudo absences and false negatives. This is an important aspect of the modeling process, and definitely therefore it's a good topic for a webinar. So we hope you all will join, and you're most welcome to join and to tell and to actually suggest colleagues to participate. So in conclusion, thank you very much, Isra, for your time, for spending a lot of time in making this presentation, and for the exchanges we had, and for your openness and help in further improving the software. Thanks to all, and see and talk to you next time.
Thank you also for inviting me.
Well, thank you. Okay, so see you next time.